So, and then here, when we're looking at the Zika markers, you can see in the infected cell lines, for the sensitive ones, there is production compared to the no virus in those same cell lines.
Production of what?
Of NS1 and S1. So that's saying that the virus is replicating and it's being produced.
And we want, that's what we want, right?
That is what we want.
Because that's what's gonna
That's
be
what's
like
killing the cells.
Perfect. Okay.
And then in
I
the
see.
resistant ones, there's nothing that shows up. So they're not replicating virus, and it's not. And then we also did a plaque assay. Are you familiar with what that is?
No.
So for the plaque assay, essentially what we do is we take the supernatant from the virus, infected plates, and in the supernatant we assume that there are viable viral particles. So like if you think about a cell dying, when it bursts open, it releases that viral particle.
When it lights,
Yeah, so we're trying to see how much virus is actually being produced in these cells, and that's what we're seeing here. So plaque-forming units per ml indicates like one viral particle is one plaque. So it forms a clearing in our plates that you can see. So we're counting the amount of clearings that there are. And so this is basically our viral output that we got from infecting the cells. So these are the sensitive cell lines. As you can see, the H23 and H1299 in particular produce a lot of virus.
And we want that, right?
We want that.
Okay.
And the resistant cell lines, they didn't produce any plaques, so we couldn't really include them on the graph. But it's just another way to prove which ones are sensitive and which ones are resistant. So now that we got this, we wanted to see, okay, there must be something in the sensitive cell lines that's expressed that's not in the resistant ones. So we looked at a panel of 6 different TIM-TAM proteins. So 3 up here are our TAM proteins, and the 3 here are the TIM proteins. And we did a qPCR to measure the mRNA expression in these cells. So in the sensitive cell lines, you can see that AXL seems to be the more prominent player here compared to the TIM or other TAMs. And in the resistant cell lines, TIM1 seems to be more of a prominent player. So that led us to think that AXL could be helping increase infection and cytotoxicity, while TIM1 may have an inhibitory effect, or in these cells, it could just be the lack of AXL. We don't know that yet.
Because AXL is a TAM protein, right?
Right.
And the TAM, and if we go, how does this correlate to this?
So we're thinking that with higher AXL, since AXL is known to mediate viral entry, more protein means more of the virus is getting in the cells,
And we
which
want-
means more of the cells are dying.
Okay.
Does that make sense?
Yeah. That's what we want. So so AXL, which is the TAM protein, was working together to make more Zika virus, right?
To kill the cells.
Yes, AXL allows the Zika to enter the cell, and once it enters the cell will make more of it and then release back it, and that will go and affect the neighboring cells.
Very cool.
So then to prove that AXL is important, we did an siRNA transfection experiment. Are you familiar with what that is? So siRNA are a small interfering RNA, and they basically interfere with translation of the protein. So right before it goes into the ribosome to get translated, this tiny RNA will bind to the mRNA. So that indicates that like the body thinks that it's something foreign, so it has to degrade it. So it'll degrade the mRNA because it doesn't like double-stranded RNA.
Yeah. So I was thinking, is it like a modifying like process of like the RNA? Is it modifying the RNA?
It's not modifying it, but it's basically tagging it for something called RISC, the RISC complex. So what RISC does is it tells the body, hey, this is not supposed to be here, we have to get rid of it. So it degrades it.
Wow. Okay.
Because RNA are single-stranded molecules, right? So when it finds a double-stranded RNA,
It says-
it thinks it is something wrong with it, so it just goes into it.
Wow.
What is that called again? The
siRNA.
And but this is an experiment. Like
Yes, yeah. So this is an experiment with AXL siRNA. So our goal is to knock down the protein level of AXL to see if that changes the viral infection. So
But
if
in
the
this
cells
case, axal expression is what is making it sensitive, right?
Mhm.
And this one does not have axal. So if we make this cell similar to that, will it prevent axal viral activity?
So you guys wanted to knock down the axle to see if the Zika virus would decrease,
Yes.
to see if then the chemotherapeutic, or well,
the therapy part of
the
it.
therapy part of treating that cancer would decrease as well.
Yes.
Okay.
So that's what we see here. These are just microscope pictures that we took of the 3 sensitive cell lines. So up here is, the top row is just uninfected cells. So this just shows the effect of the siRNA, which there really isn't an effect. They're happy, healthy growing. And then the bottom shows them infected with Zika. So in the control, you can tell that the cells are pretty much wiped out.
Yeah.
There's a lot of cell death. But when we knock down axle, in these conditions, there's still a lot of cells intact. They're living.
So axle was playing a role.
Right, yeah. And that can further be confirmed with Western blotting and plaque assay. So we saw that axle did indeed go down. The protein expression went down with an axle SI treatment. And then in the virus samples, the ones treated with an axle SI had less viral replication than the ones that weren't. Pretty significant. And that was seen in both the sensitive cell lines. We haven't done this yet, but and then again, plaque assay pretty much shows the same thing. These are the negative control. This is with the axle SI. There's a lot less plaque forming. So, in conclusion, what do we get from this? So we know that 3 of these cell lines were sensitive and 3 were resistant. We did find that axle seems to be the most prominent player in the sensitive cell lines, as proven by the transfection. And we also think that TIM1, since it's elevated here, may play an inhibitory role in cytotoxicity in the resistant cell lines, or it could just be the lack of axle. We don't know yet. We have to do further testing. So we're trying to see if we can silence TIM1, like do a TIM1 SI in the resistant cell lines, or perhaps overexpress axle, see if that affects it as well. So we think that axle and TIM1 expression can definitely be used as biomarkers to see if a tumor could be susceptible to Zika.
Wow. That is, I never saw that. Like, that is very cool.